in TV land. I'm Sarah, and today I'll be talking about gene expression and drug response. This is the first lecture in this series. I'm not sure how many there will be. Probably 3 or 4, but I'll let you know. So first of all, in the cell, we have DNA, and the DNA is sort of like the blueprint for everything that the cell makes. It lives in the nucleus. It's double-stranded. It has base pairs together. It's very, very stable. It doesn't change between cells. It doesn't change when the cell gets excited. It doesn't change ever, except when you pass on your genes to your progeny. But it's more like the static fixed reference guide, and it has to be copied into a more usable form for the cell to actually make the gene products that it's interested in making. And that more usable form is called single-stranded RNA, or messenger RNA. And it is basically a working copy of the section of DNA that the cell needs in order to make the product it's trying to make. So when we talk about gene expression, what we're talking about is the amount of that single-stranded mRNA that's floating around in the cell. If you want to make a lot of a certain protein, then you might need more copies of that messenger RNA. Whereas if you only want to make 1 or 2 molecules of the protein, then you only need maybe 1 copy of that messenger RNA. So that by making more or less of the mRNA, the cell has some control over how much of that protein gets built in the end. You can measure gene expression in a number of ways. Most popular right now are using microarrays or RNA sequencing. That will change. The technology evolves quite a lot. But basically what these two methods do is measure the abundance of the mRNA in the cell. And they do that by turning the mRNA into a much more stable version of mRNA called complementary cDNA, which is double-stranded, sort of like the DNA that's in the cell, but is really just a copy of the mRNA. It's not exactly the same as the DNA that the copy mRNA was made from. And this is because genes are actually not entirely linear. So for one gene, you might have a section. This is a sort of a standard way to draw genes. And this part here, with the big box, this section is called an exon. Sorry about the menus. And then these little sections with the lines are called introns. And then we have another exon here. So in the DNA space, you get exon, intron, exon, but the only part that the cell needs to make the protein is these exons. And the mRNA is basically a concatenation of this exon plus this exon with this intron totally removed. So instead of having a section in the book that you don't need to make the protein, it's just the parts that you absolutely need. And so when you make a cDNA copy, so if this is my mRNA, which is just the exons here, the cDNA copy will look like that, will be those sequences, and won't contain these sequences the way that the original DNA did. And the reason to do this process, to go from mRNA to cDNA, is that the mRNA is single-stranded, and that means that it's much more likely to be degraded by the cell, whereas the DNA, because it has double-strandedness, each of its sort of tender underbellies is the bases are protected from each other and from the outside world. So it's much more stable. You can manipulate it much more easily, and by touching it, it won't degrade, basically. So it's a much better thing to measure. Here are a couple of YouTube videos about the technology that's currently being used to measure mRNA expression. Microarrays are slightly old-fashioned, but still exceptionally useful and very, very widely used. And then sequencing, which is sort of the new frontier, is expensive, but works really well and has some benefits too. So I encourage you to watch those videos if you're curious about how the technology works. But I think you can understand the rest of the lecture if you just understand that what we're measuring is that mRNA measurement, not the DNA and not the amount of protein. So there's some caveats to measuring at that spot in the process. Two genes might have the same expression level, they might have the same mRNA quantity, but different amounts of protein. And that's because measuring mRNA abundance is not exactly equivalent to measuring protein levels. Certain mRNAs are more stable than other mRNAs, so they decay faster or slower, leaving fewer copies to be translated into a protein. And sometimes translation just happens at different rates for different transcripts. If you have one protein that uses a bunch of amino acids or building blocks that are rare, even if you have 10 copies of the mRNA, the process of building that protein isn't necessarily fast and maybe slower than some other gene. So it's important to sort of keep in mind as we talk about expression studies that expression isn't a one-to-one measurement of how much protein the cell is making. Okay, so next I'm going to tell you about a big project from the National Cancer Institute. And this is kind of an older project now, but it still has laid the foundation for much of the work in expression studies since they did it. And basically they collected tumor cells from 60 different patients, all types of cancer, colon cancer, ovarian cancer, breast cancer, and they transform the cells so that they grow on plastic in artificial media. So basically they kept selecting cells that would live in a mixture of glucose and some proteins from cows in an incubator in a Petri-like dish. So the cells you end up with are a little bit different than the cells straight out of the body, and you can grow them basically indefinitely. So once you have cells growing like this, it's very easy to collect the mRNA and measure it. At this time point, they were using the first generation of microarrays, which are pretty outdated by now, but they still got pretty usable data. And in this project, they treated the cells with a battery of different drugs to see which ones were effective at killing the cells. So if you have 60 types of cancer, you treat it with a bunch of drugs and you see which ones kill the cancer. So the first thing they show in their paper is that expression of mRNA corresponds to tissue of origin. And this is actually a pretty big deal. They tested the expression of 8,000 genes from each of these cell lines. You might know this, but this is out of about, this is out of about 20,000 human genes. And if you were doing this project today, you would probably test many more than 8,000. But on the first generation of microarrays, 8,000 is what you could do. So they tested 8,000. And then they compared each individual cell line to a pool of 12 of the cell lines. And they tried to pick 12 sort of diverse cell lines to be this background. So then the outcome of each comparison is how much does this particular type of cancer, does say this breast cancer from this person number 72, differ from all of the other cancers that we've seen, or sort of the background cancer expression motif. So after doing this, they got a profile of gene expression for each line, and that profile is basically what makes that cancer unique. And then they compared the different lines to each other. So this is the first figure from their paper, and they show that the expression of messenger RNA corresponds to tissue of origin. So here you see let's try green, central nervous system cancers are in gray, renal system cancers are in green, ovarian is here, down here is breast cancer, it's not labeled, but it's my favorite. Here in the pink, melanomas are here. And this is actually a pretty substantial finding because they've done all of these weird things to these cells. They've taken them out of bodies. They've started growing them on plastic. They're feeding them all the same things. Like every cell line gets basically the same diet, even though in the body it might not necessarily get the same amount of nutrients as, a breast cell might not get the same nutrients as a colon cell necessarily. The transferring them to this plastic incubator lifestyle has changed the chemical the makeup of these cells quite a bit. A lot of them start getting extra chromosomes. If you look at this cell line, MCF7, it has basically 4 copies of every chromosome instead of 2. Like, they're definitely really weird, and they're weird in ways that are not predictable. Some of them grow faster in plastic than others. This one, SCOV3, basically will double the number of cells in the population about once a week, but OVCAR3 doubles in about 18 hours. So the fact that they're looking similar to each other in terms of gene expression surprises me, because they don't act like each other. They all act like they've been through the ringer and they've come out with some sort of way to live in plastic, but it doesn't necessarily mean that they're going to look similar to other things that they came to, other cells that came from the same part of the body that they did. So this is a really big finding. And here's the citation for the paper if you want to read it. Okay. Clear this off. So next, I told you that they were really interested in screening drugs for cancer. So they found a bunch of drugs. Some of them are drugs that are on the market. Some of them were just compounds that they thought might kill cancer. And they added some to each of the 60 cell lines. And in order to quantify how strong an effect the drugs had, they measured how much protein was in the plate afterwards. So a plate that has, plate like this, that has 4 happy cells will make some quantity for proteins. Protein quantity. I'm making this up right now. Protein quantity is not a thing. Don't memorize it. Whereas a plate that has, in which the drug was not as effective and has 7 cells still alive, will have 7 protein quantity. So assuming that you started off with the same number of cells in each plate and then added the drug, you would say that the drug was more effective at killing these cells because there's less protein left. So they basically found a bunch of drugs that killed the cancer. Some of these are very surprising. Vitamin D, it turns out, kills cancer. A lot of things, if you put enough of it in a plate, will actually destroy the cells. And next they set out to find similarities in sensitivity to specific drugs between different kinds of tumor types. Okay, so the question they really want to ask is, can we predict which types of cancer will be more sensitive to drugs? We have 2 data sets that I've sort of described. One is expression data from each cell line, that dendrograph that showed the clustering of different cell lines coming back together. It doesn't have any drugs. And then we have a drug profile for each line, which is basically a measure of which drugs kill it with which efficacy. So I know, for example, at MCF7s, which is a breast line, this data set, number 2, will have something like, okay, drug number 1, Taxol, and it kills with a 90% efficacy, 90% death. Drug number 2, cisplatin, it kills with a 92% death. And all the way down. Lots of different drugs, all the way down. So I know, and I also have another, it's not erasing, another breast cancer cell line, let's say SCIBR3. I know that these 2 are related because they're from the same tissue, and I know where they cluster on that original clustering, And I know that in SCIBR3, Taxol is like 80% effective, and cisplatin is 87% effective. So now I have basically two ways to compare these cell lines. One is by the original clustering, which recapitulates where they're from, and the second is by which drugs have what kind of efficacy on them. So then we want to take these two data sets and compare them together. So we're comparing the expression profile, which I showed you before with the clustering, to the drug activity profile, those measurements of which drugs are more effective in which cells. So on the left here, this is exactly the same clustering situation that I showed before. It's just been rotated a little bit. Here's the ovarian, just to reference you. And here's a couple more ovarian down here in purple. The renals are here in orange. And these cell lines are color-coded by their tissue of origin. And then in section B here, we're showing the clustering based on the cell line sensitivity to drugs. So instead of being a tissue of origin map, this is saying, okay, well, OVCAR3, OVCAR8, and SV1C all have sort of similar response profiles to different drugs. Whereas the other ovarian, here's another ovarian cell line down here, doesn't have that same sort of response, is looking more like these neighboring cell lines and not necessarily the tissue of origin. So on the left, the tissue of origin, the colors are pretty much staying together in clusters, and on the right, you're seeing a mismatch of colors a little bit more. So things are clustering in different ways. And next they sat down to integrate these two clustering sets. And this, I know this heat map is hard to see. It's even hard to see on my screen. If you go to the reference for the paper, that you can zoom in and out and see all of the little colors pretty well. But it's still a useful thing to look at, so I'll try and describe it as best I can. What they're showing here on the y-axis is drugs being clustered together, and on the x-axis they're clustering genes together. So this is across cell lines. And basically each little spot in this heat map shows how well drug agents of this type perform in cell lines that have this kind of expression. So here they're saying a red point, like what I've just circled here, indicates that the agent over here, whatever lines up exactly with that red point, tends to be more active in cell lines that express more of that gene. Okay, let's walk through a little example that sort of illustrates what the heck this means. I think the blue is easier to understand. So I'm going to describe a sort of situation that would allow this point to arise. And what this is saying is that drugs like this, or this kind of drug, is less active in genes that express a lot, in cell lines that express a lot of this gene. So you can imagine that the drug is something, let's call it drug A, and the gene is responsible for the metabolism of drug A, for breaking it down. So if you have a little bit of drug A in here, and you have a lot of this protein that breaks it down into non-working drug A. So this is drug A, this is the gene, this is drug A breakdown product. Then if you have a lot of this gene, you're going to be breaking down this drug more and more and more, so there's not as much of it hanging around in its active form, which means that it might not have the same robust effect on the cells because it's just getting converted to X's so fast because you have a ton of this gene. The opposite situation in these red dots is also true. If you have a gene that high expression of which is correlated with high activity, you can basically make exactly the same story up. Maybe that gene here blocks the degradation of drug A, so drug A doesn't get broken down as much, therefore drug A is really, really effective in the cells. And you can sort of make up stories about all of these things. And what the heat map shows is that there are clusters of genes and clusters of drugs that have this effect together. So maybe these two drugs right next to each other have sort of similar breakdown pathways in the body, and maybe those pathways are represented in this gene cluster. But there's some systematic information here about which drugs and which genes go together, and that was a big finding for this paper. So the take-home messages from this project in 2000 is these 60 really well-characterized cell lines. They're still absolutely the cell lines that people go to first to do experiments in vitro. You can buy them on the internet for $200. You keep them in your freezer. Any first pass experiment definitely in cancer uses 1 or 5 of these cell lines. If you're studying breast cancer, you buy all of the NCI 60 breast cancer lines and do your assays. If you're creating drugs, you use these 60 cell lines. You tell people about your new drug's activity in these cell lines, and it keeps everyone a little bit on the same page. And then after you've shown that your drug works in these cell lines, you could move on to a different cell line or into animals or something. But pretty much any experiment that starts about cancer starts in one of these cell lines. The NCI 60 project showed that drug sensitivity is correlated to gene expression of a cell line, which is pretty great. That big heat map on the last slide that shows that if you have more of one gene or less of a gene, then that correlates with how sensitive you are to a specific drug. And the project set up a goal, which is basically better understanding of how drug treatments affect gene expression, and that's what I'll talk about in the next few slides.